So my name is Inder Verma. I'm a professor at the Salk Institute in La Jolla. I've been there for the last 4 decades, and I am the keynote speaker today. And my subject area of discussion today is stem cells, cancer, by using glioblastoma as an example. So what are stem cells in cancer? A cancer stem cell is a cell which exists in a cancer, which has the ability to continuously replicate and proliferate, as well as the ability to differentiate to all the lineages that are present in that particular tissue. So in the case of the brain, one would understand to think of a cancer stem cell as a cell which can continuously divide and also go on to make the major cell types in the brain like the neurons, oligodendrocytes, and for example glia. So the amazing thing about glioblastomas is that every patient who has unfortunately GBM undergoes surgery at some point to remove the big burden of the tumor, but almost 100% it is guaranteed that the tumor will come back. And sometimes the tumors come back within days and months and weeks, sometimes may take a little bit longer. Sometimes they come during the treatment and radiation that is given to the patients after that. So it is remarkable how every cell in the brain tumors, regardless of how much surgery, how good the surgery is done, will come back again and make a tumor. So how did a terminally differentiated cell that is present in the brain, like neurons, they don't divide, glia cells or oligodendrocytes, how do they go on to become a cancer stem cell? What I'm going to point out is that when there's an oncogenic insult, meaning when their genes which have the ability to cause cancer get into one of these cells as a mutation, undergoes a mutation, it then de-differentiates or reprograms a terminally differentiated cell back into stem cell. After all, a neuron or a glia or an oligodendrocyte originated from a stem cell. And what has cancer done to them is that when oncogenes are mutated or undergone a change, they reprogram or de-differentiate back into a stem cell. And if you recall, that is now a very well accepted fact, what we call the iPS technology, where a terminally differentiated cell can go on to become stem cell, and in fact, in some cases, they can then go on to become any cell type in the body. So, what I want to emphasize in my talk is that this is a remarkable situation where we now understand how glioblastomas are formed. So now that we know, our job is to prevent these cells, to either prevent them to differentiate or to continuously proliferate. So not only that, we discovered something else along with it. And that was that brain is highly vascularized. In fact, it's probably the most vascularized organ in the body, not surprisingly, because it's continuously functional, needs metabolism, needs food. So why not try to block blood vessels going to the tumor? Because that will not take the food and therefore the tumors will die. But unfortunately that didn't work. Because what happened is these tumors actually further on transdifferentiated to become a blood vessel. So normally you have a process of blood vessels, and you can use inhibitors which prevent the formation of blood vessels. But these tumors actually went on to become blood vessels in a different manner, by which all these inhibitors of blood vessels were ineffective. So the tumors are pretty smart because they only have one thing to live in life, is to grow. So not only they grow, they also transdifferentiate. So I will try to talk in my keynote address how this process functions, what's its importance to understand the biology of cancer, how we can take advantage of this, because the very fact by which tumor actually becomes so malignant also offers us the Achilles heel that we can stop that malignancy by letting it growing or to differentiate or to transdifferentiate. So that's what I will try to then bring together, and how basic biology of understanding stem cell can eventually lead us to understand how cancers were formed and eventually therapeutics, which of course has wider implications because all cells in one way or the other have origins in stem cells. So by understanding this principle, we actually will also understand other principles of stem cell, including cardiomyocytes, hepatocytes, and muscle cells. And of course, they also then offer us the opportunity to intercede with the progression of the disease. Because the technology we developed here are the same technologies which can be used for other things. For example, cerebral tumors, for cerebral hemorrhages, and other kinds of diseases which are linked to the brain. So that will be the general theme that I will be covering.